Yes, so you can see my screen already, I assume. Thank you very much for inviting to this conference. I'm going to talk about the evolution of virulence in the commensal bacteria Escherichia coli. And this is work that Julie Marin mainly did with myself, and I'd like to acknowledge collaborators on this work, so for Norouzian and Olivier Clermont for the data, and Olivier Tenaillon and Eric Denamur for the conception of the project. And this was funded by a CNRS grant. Okay, so E. coli is at the same time a commensal, but also a major pathogen. It lives in the gut of almost all humans. It also lives in other animals, wild animals, farm animals. It can be found in the environment, in water, for example. And its typical habitat is the gut, but it can also cause very rare infections. For example, intestinal infections, at a rate of about 10 to minus 2 per year. Urinary tract infections are also quite common, and more rarely bloodstream infections at the rate of 10 to minus 4 per year. And all of these infections are rare. Since everyone carries E. coli, they still cause a considerable burden. So there's of the order of 1 million deaths from E. coli infection each year in the world. And so I'm going to focus more specifically on extra-intestinal virulence. So that is urinary tract infections and bloodstream infection. And this type of virulence is linked with the presence of virulence genes. So these genes are, so they belong to the accessory genome. That is, they can be absent or present in strains. And this table shows results from a study where they compared commensal strains, so strains found in the stool samples of healthy individuals, compared with strains found in bloodstream infections, which are the ones that most often we study because they are systematically sampled as opposed to the one in commensalism. And this is a list of virulence genes that are more or less associated with being found in commensalism or in clinical samples with a p-value on the right here. So I'm highlighting several of these genes here. So the ones with a significant difference in commensal strains and clinical strains. And you can also compute a virulence score, which is the mean number of these virulence genes in this list that were found in the two settings, in the two type of samples. And this also has a very significant difference in commensal strains and clinical strains. And one complication with this kind of analysis is that E. coli has a very strong population structure. So this is the phylogeny of the core genome of E. coli, so all the genes that are found in most bacterial cells. And you can see here that this phylogeny is heavily structured into what are called phylogroups, with phylogroup A in blue, B1 in green, and B2 in red being the most abundant in those healthy volunteers. And this can complicate the analysis of the effects of individual genes because these genes are associated usually with phylogroups. Nevertheless, when you look within, for example, phylogroup B2, so this is this other column on the right, some of these genes still come up as significant. So even within phylogroup B2, they do have an effect on virulence, so on being found in infections versus in commensalism. And if you can actually translate this type of data to an effect of each gene on the probability to cause an infection. And if you look at these genes, which I showed have a significant effect, even within one phylogroup, they translate into a plus 20% to plus 30% increase in the probability to cause an infection. And so this is a list of selected genes, and this is also being phylogroup B2 strain is also associated with a higher probability to cause an infection. So the evolution of these virulence genes is potentially of public health interest. For example, if one of them increases in frequency in the population, this would translate in potentially many additional infections worldwide. So for example, this IUCC gene, if it increased by 10%, this would translate into 20,000 additional bacteremia per year. Many of these bacteremia leading to death of the host. So the question I wanted to ask is what drives the evolution of these virulence genes? And the one first answer is the evolutionary cost of causing infection. And usually people think of this in terms of if you cause the death of the host, you impede onward transmission of the pathogen, which is negative from the pathogen fitness point of view. So this is an old theory, and this explains why, probably why most of these bacteria are commensal. You could also argue that if you cause a more benign infection, like a urinary tract infection, nowadays people would often take antibiotics, and this would get rid not only of the bacteria in the urinary tract, but also in the gut, and this also probably suffer some form of cost from this. But this cost, as the infections are quite rare, is probably quite small as well. And there's a small body of literature documenting positive effects of these virulence genes in commensalism. So on the left, this is a study with longitudinal follow-up of infants in the first year of their life in Sweden, and they characterized the dynamics of E. coli. So there's a constant turnover of strains in the gut, and they characterized the strain by whether they were transient, so they stayed for a very short time, or intermediate, so for 1 month to 11 months, or long-term, meaning that they reside for more than 12 months in the gut of these infants. And what they see is that as a function of the presence of different virulence genes or virulence islands, so the ones which carry, for example, the islands are also more frequently long-term colonizers, so they might enable longer persistence in the gut. And there's also some experimental work. So for example, here where they show that when they delete some of these virulence islands, the bacteria are less good colonizer in mice. So here we wanted to investigate that a bit further, and we focused on one specific virulence genes, which is actually an island, so a group of genes, so the Yersinia pestis high pathogenicity island. And we focused on this one because it's relatively uncoupled from the phylogroup structure, so not totally, but relatively. And also it increased in frequency, at least in France in recent years, as I will show afterwards. This is a piece of several genes forming like about 34,000 bases, and it's involved in iron capture, so it synthesizes siderophores, so molecules that bind to iron, and also enables the transport of the siderophore iron complex into the bacterial cells. And so we used to study the evolution of this specific virulence island, so we used about 400 E. coli genomes that were sampled from the stool of about 400 healthy volunteers, mostly in Paris, and from 1980 to 2010. So this table shows the sampling year and the number of genomes. So a lot of them come from recent time, but we still have decent sampling from 1980. And it's quite important that this comes from commensal. So these are E. coli from in a commensal context, because this enables to look at the evolution of virulence genes, whereas if you look at clinical samples, you bias upwards towards strains that are more virulent, so it's difficult to control for this bias. And so, as I said, the HPI increased in frequency in 30 years. So on this graph, you can focus mainly on the black line, showing that it was about 30%, 35% in 1980, and then increased in frequency till 2010. And this increase is significant even when controlling for the phylogroup structure. And this results in an estimated selection coefficient of 0.003 per year. Another way it says the same thing, that it increased in frequency. And so this increase could have caused a few hundred additional bacteremia per year and thousands of urinary tract additional infections per year, if indeed the effect of the HPI is as estimated from the comparison of clinical and commensal strains. And this is just to show that, as I said, there's always this problem that there's a lot of structure, and concomitantly, we also had changes in the frequency of different phylogroups of E. coli, the most notable being the decrease in phylogroup A and the increase in phylogroup B2 and D. B2 and B1, sorry. well, B1 is not so apparent, mainly B2. But as I said, the increase in HPI is still significant even when controlling for these changes. So the questions we wanted to ask is, when did the HPI emerge in E. coli? And the hypothesis we had at the beginning that maybe the HPI emerged quite recently and then increased in frequency as we observed in France. How does the HPI spread? And what is its ecological advantage? And for the first question, it is known that the HPI comes from Yersinia pestis, and it jumped to E. coli and wanted to date that more precisely. So Julie looked in the literature, and she found this recent paper from Nicolas Rascova and collaborators who investigate the evolutionary history of Yersinia pestis and link them with the history of different plague epidemics in Europe and the Middle East. And in this paper, precisely, they had a new strain, which is called Gochem2, which is a strain of Yersinia pestis from Neolithic farmers in Sweden, dated 5,000 years before the present. And so she looked into these strains and isolated the HPI when it was present and when the quality of the genome was good enough to be able to isolate it. And she did a molecular clock analysis. So a phylogeny of HPI that incorporates both the HPI from our E. coli samples and in gray, the Yersinia pestis HPI. So the x-axis now is time with these old HPI from sample from Yersinia pestis isolated in this in this Swedish Neolithic farmer. And when you do that, we can date the acquisition of the HPI from about 4,600 years before the present. What was and the analysis was actually quite robust, because even before she incorporated these old sequences, she estimated an acquisition date that was quite similar to this one. So this is not totally constrained by, well, actually not constrained by the fact that our older sequence is about 5,000 years old. So it was quite robust. So the HPI emerged more like a longer time ago than what we thought originally. I mean, we didn't have any argument in favor of a recent emergence, but so they emerged like about a few thousand years ago. And we wanted to see if in the genomes we could find evidence of selection for the HPI since it was introduced. And for that, we looked at the phylogeny of the core genome labeled by HPI presence and absence. So absent in blue and present in yellow. And the slight difficulty with this is that introduction of HPI is very recent compared to the whole evolutionary history of E. coli, which we estimate with very large confidence intervals, because there's some difficulties with molecular clock at this level, but about 1 million years ago for the MRC of everyone. But regardless, the HPI introduction is very recent. So we try to look at what happens when you cut this tree and you look at these recent clays. So here's the last 4,600 years of evolution, so when the HPI was introduced at the bottom, and to see whether we could link the diversification of these subclades to the presence or absence of the HPI. And really, most of the diversification took place in very recent time. So this is zooming in and looking at the last 100 years of evolution. And now you can start to see these clades diversifying. And so when you correlate the size of these subclades, like as a simple first approach, so the y-axis on this graph is the size of the subclade, and the x-axis is the frequency of the HPI within the subclades. So the bubbles are proportional to the number of occurrence of this particular instance. So this big bubble is the singletons, which are non-HPI, for example, and this one here are the singletons that are HPI. And you see a correlation between these two things. So that it seems like the diversification is associated with HPI. This is quite significant. And this is when looking at all sequences, but this pattern is also found when you, for example, exclude phylogroup B2, just as a way to control that it's not this phylogroup which is diversifying more and also has more HPI. So if you exclude it, you still find a similar pattern. And what we did next was to try to actually fit a model to that. So we we tried to look at whether the diversification of these subclades was associated with the presence of HPI. So we used models that are used in comparative approaches where the branching of these trees is described by a coefficient lambda, which differs whether you are non-HPI, so lambda 0, and lambda 1 when you are HPI. And then you have transitions from acquisition of the HPI or loss of the HPI. And you can actually fit this model with some kind of composite likelihood that takes into account all of these subclades at the same time with the same parameters. And when we do that, we find that the branching rate, so the two first histograms are the two branching rates, so 0 when there is no HPI, and 1 when there is HPI. And you see it's much larger when there is HPI. And the gain of HPI is here, and is smaller than the loss. So you lose HPI at a higher rate than you gain it. And so we are aware that this analysis is a bit, so we take this branching process model forward in time, and we apply it to things that a phylogenetic tree that is represents a sample from population, so more maybe closer to a coalescent type of assumption. So we want to do simulations, which we didn't have time to do, to check that this method actually tells you what you want it to tell, that there is, when there is selection for an element, you see it as a more rapid diversification, as we see here. So this is evidence that maybe the HPI allow faster diversification, so was selected on these recent timescales. And as just another small note is that there's a type of analysis where you just look at the whole phylogeny and you treat absence and presence as a binary character. And if you do that and you look, for example, at phylogroup B2, you would infer probably that B2 acquired the HPI quite far in the past, because this is a more parsimonious explanation, and then lost it a few times. But when you look at the sequence of the HPI, you see that it's not what happens, because it's much more recent. So actually what happened is that the HPI spread very rapidly within phylogroup B2. So the third part is about the ecological advantage of HPI. So this was try to look a little bit further into the ecological mechanism, the selective advantage. So for this, I reused data from so this paper, Nourouzian et al., where they collected, so I mentioned this data already. So they collected the stool samples of infants in the first year of, at different ages, and they characterized the colonization dynamics. So many infants are not colonized initially, then they are colonized by E. coli, and there's a turnover of strain, and they can be sometimes doubly infected. And as I said, they look into, they categorize strains as with different residency times. And we went a little bit further by explicitly fitting a model of strain colonization and strain turnover to this data, where individuals can be empty, so they are not colonized, then colonized by a strain without HPI, doubly colonized with one without HPI, doubly colonized with 0 and 1, etc. So you represent all states of single and double colonization. And you also model loss of these strains. And when we fitted this model to the data with a Markov chain model, we were able to infer these different parameters. So really looking into the ecology of strains with and without HPI, and what we find, so these are the estimated rates. So the colonization, ability to colonize, was similar for the two types of strains here. So this is the ability to colonize empty hosts, and these 4 coefficients in orange are the ability to colonize hosts that are already occupied. And although these 3 are similar, this one, there's a weird pattern whereby it's very, so I can't fully explain this, but it's very, there's a very fast rate of colonization of individuals colonized by a non-HPI strain by another one without HPI. But the main difference comes between the 2 strains come from the ability to persist. So the rate of loss. And in single colonization, the rate of loss is much faster when you do not have the HPI than when you have the HPI. So I think this is the main, so if you integrate all of this and you simulate the model, the main selective advantage, so overall HPI is very strongly favored, and the main selective advantage comes from this. So the fact that you can persist much longer in single colonization. And for dual colonization, so again, these three types of dual colonization have similar rates of loss. And this one is so when you have two non-HPI strains, one of them is lost very rapidly. So I can't really explain why when you have this, you have like a really fast turnover of an additional non-HPI strain. I think the main message is that in single colonization, HPI is a strong advantage. And this results in a huge selection coefficient actually. So if you take these numbers for, if you assume that in the whole population these numbers are valid, then you would predict that HPI invades quite fast the population, actually much faster than what is observed. Okay, so the conclusions is that, so in France, we see in commensal strains that there is a rapid increase in frequency in the HPI in 30 years. And so this increase in frequency will result in a number of excess bacteremia that is of the order of a few hundreds. So that's relatively, can appear relatively modest. This is only one gene, so I think it would be interesting to look into other genes and see how they evolve. And this will also cause thousands of additional urinary tract infections, which are more benign, but nevertheless also a public health problem. So we dated the introduction of HPI to about 4,600 years ago in E. coli. We see in the genomes that in the phylogeny of the core genome that the high frequency of HPI was associated with a recent diversification, which could suggest also selection for HPI over timescales of a few hundred years. And we see in the data at a smaller time scale, so this colonization data, there is a very strong selective advantage in colonization that is linked with the persistent in single colonization. I'm not quite sure why, I mean, what would explain a recent selection for HPI, but one hypothesis is maybe if you have improved hygiene, you would reduce opportunities for colonization for E. coli, and therefore it would be interesting to reside for a longer time, to persist in a longer time in a gut once you're there. So maybe this could explain why the selection coefficients in favor of HPI had changed in recent years and favoring HPI more than it used to be. And I will stop here. Thank you for your attention.
Okay, thank you. Any questions from the chat? Well, please type your questions. Any questions from people speaking? If not, I have a question. So how does this inference depend on how much data you have? I mean, how basically, you know, how much data do you need to get these kinds of, to reach these conclusions? And would it be hard to do it for other problems?
I think the main limitation to obtain this data is that not so many people study commensal strains because they are more difficult to sample. So to study the evolution of virulence in commensal bacteria, you need these commensal strains. So how many? I'm not quite sure which part are you.
No, I mean that's the sort of, you know, you know, as you say, it's an unusual topic. So I was sort of thinking about what are the main limitations in getting this kind of data, and then yeah.
Yeah, I think I guess one of the reasons why people didn't look too much into it is that if you look at strains from clinical samples, then you wouldn't see an increase in virulence genes, probably because these strains are heavily overrepresenting virulence genes already. So it's really hard to detect trends in these.
Okay, we have another question from Guillaume Achaz. HPI increases the diversification rate by increasing the birth rate or reducing the death rate?
Yeah, that's a good question. And actually, I think they are a little bit confounded. So we use a model where we constrain the death rate to be 0. So diversification is birth really, because the death rate, the extinction rate is difficult to infer precisely. So in short, we don't know. And also, I think this model with birth and death, I'm not sure is super relevant when you're looking into the genealogy of a sample. So I think the simulations I was mentioning will answer that.
Okay, anybody else? I don't want to sort of...
I have a question. So the fact is that all your samples are are from today or very close to of today, and doesn't make a bias in that you see all the diversification as quite recent in the last 100 years. Is it really? I mean, are you really sure of this, or is it because you see only the recent things? If the HPI is something that goes in and out. You're not going to see all the thing that didn't, the lineages that didn't go to the present. You don't see them.
Yeah, so I think it's a good question, and we need to think about like how the current sampling, I mean, the fact that we sample bacteria in the present affects this, but I don't think I find it difficult to see how this sampling effects could differentially affect strains which have the HPI versus which do not have the HPI. What could be possible maybe is that if you have a structured population, so you have strains that are more specialized to water and strains that are more specialized to sample, maybe many of the singletons you see, so those very long branches, so the strains that are that do not have a cousin or a sister in the past few hundred years. So these could be non-adapted to humans, so they could be adapted to water. And then, so this, what I showed would be just evidence of an association between HPI and being a human commensal. So I think there could be sampling effects in that respect with regard to the maybe a structure of E. coli into different habitats.
Okay, anybody else? Questions?
Okay, I have a last question. So in your model, you're not looking at all at. I mean, you're not looking much at population dynamics. I imagine that for E. coli, the fact that the number of humans have increased a lot and the connectivity between humans has increased a lot could also be a factor in what you see. How much is going to affect your models?
Yeah, so that's a good question. And so Julie looked into the effective population size as a function of time from these samples. So using the Bayesian skyline plot in the BEAST software. And we see, so there's no like strong pattern. It's kind of stable. There's like a small increase in the recent past of this effective population size, but it's quite difficult to interpret. I'm not even sure it's significant. But yes, this would probably affect in some ways the coalescent rate from the approach we took. So based on just inferring the effective population size with coalescent rate, we didn't find much of an effect.
Okay, thanks a lot.
Okay, thank you. And I realized I forgot to clap, so I'll clap now. So thank you very much, François. And